Hello friends, welcome to the Knowledge Club Online. Now I am going to discuss about the penetrance and expressivity. The percentage of individuals that shows a particular phenotype among those capable of showing it is known as penetrance. Let us take an example of a polydactyl in human, which is produced by a dominant gene. Homozygous recessive genotype does not cause polydactyly. However, some heterozygous individuals are not polydactylous. If suppose 20% of heterozygous individuals do not show polydactyly, this means that the gene has a penetrance of 80%. Degree of expression of a trait is controlled by a gene. A particular gene may produce different degrees of expression in different individuals. This is known as expressivity. Different degrees of expression in different individuals may be due to variation in the allelic constitution of the rest of the genome or to environmental factors. Thus, the terms penetrance and expressivity quantify the modification of gene expression by varying environment and genetic background. They measure, respectively, the percentage of cases in which the gene is expressed and the level of expression. What is the incomplete or variable penetrance? Incomplete penetrance is not the same as variable expressivity. In disease with a variable expressivity, the patient always expresses some of the symptoms of the disease and varies from very mildly affected to very severely affected. In autosomal dominant diseases with incomplete penetrance, the person either expresses the disease phenotype or he or she doesn't. Incomplete penetrance and variable expressivity are phenomena associated only with a dominant inheritance, never with recessive inheritance. Now, this is the example, a perfect example of incomplete penetrance. The disease is passed from the father to 3 and to the son, passed from the 2-3 to the son 3-5. This never happens with X-linked traits. The disease occurs in 3 consecutive generations. This never happens with recessive traits. Males and females are affected with roughly the same probability. However, 2-1 does not express the disease. He must have inherited the mutant allele because he passed it on to the 2 children, 3-1 and 3-3. Is a classical example of incomplete penetrance. He has the allele for a disease, but he does not express it. The factors affecting gene expression. The many genes have consistent and regular patterns of expression. Some genotypes, the phenotypes may vary. Gene-gene interactions, gene-environment interactions, that is the temperature and age-dependent interactions. This is the egg colors of endangered Hawaiian poa bird is controlled by a single locus, BLU, where the B allele is dominant to the small b allele. As part of the recovery strategy, a pure breeding blue egg bird (BB) from one island is crossed with a pure breeding white egg bird from another island. Because all the offspring are BB heterozygous, they are all expected to show a uniform blue phenotype like that of the BB parent. However, if penetrance and or expressivity vary, the 3 patterns of variation that differ from expectation are possible. If penetrance varies, the genotype sometimes does not penetrate through to the phenotype. Some eggs are blue just like those of their parents. Others are white like those of their parents. Despite presence of the BLE, lack of penetrance in sometimes used to explain individuals whose phenotypes do not reflect their inferred genotypes. That is a dominant trait that appeared to skip a generation in a pedigree. If expressivity varies, the BB genotype will not be expressed uniformly in the phenotype. All eggs are blue, but the exact shade of blue varies among individuals with the same genotype. Variable expressivity is frequently attributable to environmental factors or variation at a gene loci elsewhere in the genome. If expressivity and penetrance both vary, there may be continuous gradient of phenotypes between white and blue eggs. Note that lack of penetrance can be considered as an extreme form of expressivity, in which the range of expression includes non-expression. Now come to the temperature dependent inheritance. Various factors, both genetic and environmental, can affect the color of Siamese cat. As the points and body color are temperature sensitive, an important factor in the length of pigmentation is the amount of heat loss from the skin. Color is affected by climatic conditions. For instance, in Australia, the Northern Territories' hot climate has a dramatic effect on seal point Siamese, whose points become brittle with cream and seal hairs intermining. In contrast, Stars and other cats kept in an outdoor cattery in Tasmania winter cold have dense seal points and are darker in the body. Theoretically, if we keep our Siamese warm, especially seal points, the lighter their body color should be, but not too warm, as an elevation in temperature can cause brindling or loss of pigmentation in the points. Often, see after the cat has been ill, Siamese grow out darker on the body after surgery, and white hairs can appear on the points when the follicles are damaged. Age is another factor, and the sites of intense pigmentation appear of the shoulders and hips where the skin is closest to the underlying bone structure. The lighter the color, the more sensitive to heat. Chocolate is more sensitive to heat than seal, means black. Hence, the difficulty in breeding a chocolate point with perfectly even points. The temperature alone affects color is an oversimplification, as there are many other genetic factors including modifiers and polygenes. This lecture may be helpful to you. Thank you.